Hello, I'm Kelly Diamond, and on this project, I'm working with Ron Kwan and Murat Maga to measure craniofacial variability in zebrafish using computational anatomy. And as a morphologist, I'm interested in why animals are shaped in all the ways they are shaped. And a huge contributor of this is genetic makeup and the influence of genetic variation on phenotype. One way to quantify the relationship between genotype and phenotype is to compare the phenotype of known genetic mutants and their wild-type controls, or their wild-type siblings. And the MSB lab at University of Washington has developed an approach for decoding the spatially variable phenotypes of the axial skeleton. Their FishCut program isolated individual vertebrae and their associated structures along the length of the axial skeleton, and then analyzes the bone mineral density throughout their phenotype. And this is really important because one of the mutants that they examine with this technique is the BMP1A mutant, which has a human ortholog, the BMP mutation, which is associated with brittle bone disease. And you can observe the differences between the mutant on the bottom and the control fish on the top. The mutant fish has severe deformities in their ribs and thicker vertebral centra along the length of the body compared to the wild type sibling fish. And also the mutant is shorter in length than the control fish. While these are all very interesting, one area of the anatomy that this study did not look into was the fish skull. And that's because fish skull are a bit more complicated. So to start this project off, we started with the micro CT scans of the skulls of BMP1A mutants and their wild type siblings from a single family of zebrafish, with the goal of quantifying phenotype between these two groups. And to do this, we're using a computational anatomy technique called atlas building, which iteratively finds the virtual space for a centroid of the population. And to find this centroid, we use a series of registrations that first puts two images that we are comparing into the same space, and then minimizes the difference between the shapes being deformed and the reference image until we end up with an average model or atlas image. And just to give you an example of the types of registrations that we use, here I have a moving image of a zebrafish here in pink, and it's transformed towards a reference image of another zebrafish here in blue. And first, the rigid registration uses only rotation and translation to put the moving image in roughly the same space as the target image. Second, the affine registration uses rotation, translation, as well as scaling and shearing to make the moving image have roughly the same dimensions as the target image. And finally, the deformable registration allows every point in the target image to move in order to minimize the difference between the moving and target images. And so what we do is we iterate through all of the images in our sample in order to create an average atlas image. And for our specific study, we used the answer package in R, and we only used the wild type fish to build our atlas because we wanted to get a baseline of what the wild type anatomy was in order to compare it to our mutant fish. However, before we can quantify phenotype, we need to test that our atlas is doing a good job of representing the anatomy of interest. To test the atlas, we use otoliths or ear bones of our zebrafish. These videos show a manual segmentation on the left and a segmentation created from our atlas on the right of the otoliths from one of our mutant fish. To orient you to the head of the fish, the head is facing down, just like in the diagram in the upper right corner of the slide, and we are looking down from the dorsal view of the fish as the video starts to play. We can see that both the manual and atlas segmentations do a pretty good job of isolating each otolith. And when we plot the volumes of the manual otoliths and the atlas otoliths that are segmented for each fish, we get a very strong correlation. This indicates that overall the atlas does a decent job of segmenting the otoliths. Just to further point this out, the mutant fish are in orange and the wild type fish are in gray. We have three clusters of points here because there's three different otoliths that are different sizes. And we can see that overall we have this tight correlation, although the mutant fish do seem to have larger otoliths than the wild type fish, which is something that we've reported back to Ron. So now that we have a decent atlas, what we want to do is quantify the phenotype. And the traditional way to do this is to use landmarks, which are homologous points on every specimen in our sample. However, due to the sparsity of traditional landmarks and the subtleness of the mutant phenotypes, traditional landmarks may not capture the phenotypic differences in our sample. So here, I propose using a pseudo-landmarks as a first pass to identify areas of the skull that vary the most among mutant and wild-type fish. By pseudo-landmarks here, I mean landmarks that are not homologous, but instead are evenly spaced across the surface of each fish skull. I want to point out the SlicerMorph logo here in the bottom right corner. Anytime you see this logo on the remaining slides, it indicates that a SlicerMorph package in the open source 3D Slicer app was used for that step of the process. So the first step of our pseudo-landmark pipeline is to generate the pseudo-landmark points. And we did this using the pseudo-landmark generator in SlicerMorph. Here we were able to generate, using the surface geometry of our atlas, about 500 pseudo-landmark points across the surface of our atlas. Now that we have the points on the atlas, we need to transfer those points from our atlas to each individual specimen in our sample. And there's two ways that we can do this, which we are actively exploring to determine which method works best on our pipeline. The first method uses the same R package we used to build the atlas, answer, and requires volumetric data, such as those you get from CT scans. The second method is implemented in the alpaca module of SlicerMorph and uses 3D models instead of volumes. So for this step, we have an additional step of creating 3D meshes from the original CT scan volumes. Both methods use a series of registrations and transformations to get the points from the atlas into the target specimen space. So here I have the original atlas with purple points showing you the pseudo-landmarks from the atlas. And in the second video, I have the answer and alpaca pseudo-landmark points on one of our BMP1A mutants. And we can see that both methods transfer the pseudo-landmark points pretty well to our samples. So now that we have all of our points, we want to see if the phenotypes differ among our groups or among the methods used to transfer the points. And to do this, we ran a generalized progression analysis on the combined data sets. In this principal component plot, I have the mutants in orange and the wild-type fish in gray. The circles represent specimens from the alpaca method, and the triangles represent specimens from the answer method. The larger shapes represent the average image for each group and method. Both methods show separation in the morphospace between the mutants and the wild-type fish. However, alpaca may be a little bit better at separating these two groups because the separation is across multiple principal components. So what I mean by that is you can see that the circles here are separated both along PC1 and on PC2, whereas the triangles are mainly only separated across PC1. So for now, it looks like alpaca might be a better pipeline for this particular data set. However, but what we really want to know is how do these groups differ in shape? So here I have the images for the first two principal components. Each dot represents one of our pseudo-landmarks, and the size of the dot represents the variation among our samples at that pseudo-landmark. The green vectors coming off of each plot show the magnitude and direction of each pseudo-landmark for a given principal component. So I'm going to play the first principal component, and it's a little bit hard to see with this quick of a spin, but the length of the vectors is the longest in the lower jaws above the eye and where the pectoral girdle attaches to the body. It's a little bit easier to spot the larger area of variation in PC2. And both the size of the dots and the length of those vectors are biggest and longest, immediately posterior to the eye, and again, where the pectoral girdle connects to the body. So the next step for this pipeline would be to examine these areas of interest in more detail, perhaps with more traditional landmark methods focused specifically in these areas. The goal of this project is to develop a pipeline that will allow researchers to scan complex phenotypes to identify areas of greatest variation. In the current dataset, the alpaca pipeline seems to slightly outperform our answer-based method, though the alpaca does require an extra step in building 3D meshes from the volumetric data. Moving forward, we plan to test both pipelines on additional families of mutant zebrafish to assess which methods would work best for general use. I'd like to thank the members of the Alpaca and MSB labs, and please reach out if you have any questions about this process. Thank you.